Welcome and good afternoon to those here on the West Coast. I'm Melinda Kliegman, the Public Impact Director at the Innovative Genomics Institute, which is a partnership between University of California, Berkeley and University of California, San Francisco, founded in 2014 by Jennifer Doudna, with the goal of using genome engineering to solve some of humanity's greatest problems, while also advancing public understanding of the benefits of genome engineering. The topic of discussion today is the Alliance for Therapies in Neuroscience, a recently announced long-term research partnership with Genentech and its parent company Roche Holdings to accelerate the development of new therapies for neurological diseases. Today, I have with me several scientific leaders from UC Berkeley and UCSF, and we will be discussing the role of CRISPR-Cas technologies from bench to the bedside to treat neurological conditions. If you have any questions for the panel, please use the Q&A function in Zoom or through the chat function on YouTube, and we will have time at the end to address those questions. So the excellent panel of experts we have with us today are Dr. Jennifer Doudna. She is the Li Ka-shing Chancellor's Chair and a professor in the departments of chemistry and molecular and cell biology at UC Berkeley, and the founder and president of the Innovative Genomics Institute. Her groundbreaking development of CRISPR-Cas9 as a genome engineering technology with collaborator Emmanuel Charpentier earned the 2020 Nobel Prize in Chemistry. Our second panelist is Dr. Stephen Hauser. He is a Robert Fishman Distinguished Professor of Neurology at UCSF. He is also the director of the Weill NeuroHub at UCSF. The Weill NeuroHub is an umbrella organization that links the clinical and basic neurosciences to accelerate research against neurologic diseases. A neuroimmunologist, Dr. Hauser's research has advanced our understanding of the genetic basis, immune mechanisms, and treatment of multiple sclerosis. And our last panelist is Dr. Ehud Isacoff, or Udi, who is the Evan Roche Chair in Neuroscience and director of both the Helen Wills Neuroscience Institute and of the Weill NeuroHub East at UC Berkeley. Udi also leads the Berkeley Brain Initiative and is a pioneer in the field of optogenetics research. His laboratory at UC Berkeley conducts research applicable to neuronal diseases, including retinal disease, to restore neural function. So welcome, Jennifer, Steve, and Udi. Excited to have you all here today and to have this conversation with you. So I was wondering if Steve, maybe you could kick us off by setting the stage. Can you give us a brief background of the state of the current field of neurological disease research? Where is this partnership picking up from?
You're muted,
I think
Steve.
you're on mute, Steve.
Excuse me. Good afternoon, everybody.
Good afternoon.
Now you hear me. Well, I'm so pleased to be here and humbled to kick this off, but delighted to. All of us are tremendously excited and hopeful about the new partnership between NeuroHub, the IGI, and Roche Genentech to advance understanding and therapy for patients with neurologic and psychiatric diseases. As we all know, this is an area of enormous suffering, of enormous cost to the world of healthcare. More than 1 billion people develop a neurologic or psychiatric disorder each year. And with the aging of the population, we are seeing and will continue to see an explosion in age-related neurologic and psychiatric diseases. And I would highlight, maybe in just a slightly biased way, that aging is predominantly, from a medical perspective, a neurologic problem. You mentioned that I'm a neuroimmunologist, which is correct. And for more than 40 years, I've been studying inflammations of the brain, and especially multiple sclerosis. And MS is a particularly challenging, but also instructive, complex disease because it's not a single gene disease. There are more than 230 genes, most of them active in the immune system that contribute to risk, but most of the risk is also environmental. And the explosion in the prevalence of multiple sclerosis, which is now the most frequent crippler, apart from trauma, of young adults, is certainly due to environment and not genetics, because our genes don't change that fast. MS has two forms. There's a relapsing form with attacks, and then a progressive form with slow disability to a wheelchair-bound and then bed-bound state. Relapsing MS is now largely in the rearview mirror, thanks to advances. And we now, since 2017, have incomplete but very promising effective therapies for the progressive form of the disease as well. And the science that has connected the translational work that has made these advances possible points the way to final answers for MS as well. How did this happen? I think there were three big points. One was development of a better preclinical model for the disease. Second, advances in cellular and molecular biology that made it possible to truly understand what's happening in people and to bring that back to the models and vice versa in an iterative fashion. And third, also, a large dose of good fortune, but something that was enabled by bringing people together across disciplines. Now, maybe in the last minute or so, I might say, Melinda, that neuroimmune diseases may be low-hanging fruit. They're a little bit easier than some of these other challenges, but there has also been recent progress with other brain disorders that we thought we never really could get our hands around. Circuit-based disorders, epilepsy, migraine, for example, have seen wonderful progress. And I'm especially excited about the neurogenetic disorders against which we are just beginning to make real inroads. Spinal muscular atrophy, congenital blindness, inherited amyloid neuropathy, and as many of you know, thrilling, very recent preliminary data in genetic forms of Lou Gehrig's disease, or ALS, that might point the way to real treatments, at least for some people with these diseases. There are so many aspects of complexity that we're going to have to deal with these disorders. And I think our biggest challenge is to reduce the complexity. We've learned that degenerative brain diseases are not single entities, but they're different problems in different people, and maybe even have different biological pathways that are driving tissue damage at different stages of disease in the same person. So our challenge to reduce the complexity, I think, gives us our greatest chance of success in the near term. And perhaps disorders that have the phenotypes of common disease, but we know are caused by single genes, might be one route to going after reducing complexity.
Thank you for that, Stephen. So maybe I can turn it over to Udi as co-director of the Neuro Hub. What do you think this new partnership opens up, and what sort of unmet need do you think it helps to address?
Yeah, thanks. I think that the need is huge, as Steve was describing. The problems are fantastic, right? I mean, they just boggle the mind. Steve mentioned many diseases, and two of the ones that are so big in neurodegeneration, he skipped over: Alzheimer's and Parkinson's, which are emblematic in many of the same ways that he described. Immunity plays a big role, right, in them, although different aspects of immunity than what he described for MS. Many genes are involved in the genetic variants of them, although they only maybe account for less than a fifth of the cases of Parkinson's. And they may account for actually a majority of ones of Alzheimer's, except there's so many of them that their interactions are very, very complex and they're just not understood. And so the result of this is that, while we've made real inroads in some of the conditions that Steve described, we've been stuck, really terribly stuck for decades on some of these others. Even when we had unusual cases with clear genetic association, it's been a struggle. And for the ones where it's many genes playing a role, it's very, very hard. So the question is, how do you get at this? This is a problem for us as scientists at our two universities. It's a problem for pharma and biotech, like Roche and Genentech. And the beauty of it is that there are new kinds of approaches that maybe could break through. And one of the places where there's enormous excitement now is in what CRISPR technology is going to be able to do. This is why we're so excited about IGI being a partner in this. We've become very, very used to the dream, right, of that one day a genetic disorder can be repaired by editing the gene, right, that is responsible. And in some cases where it's a very clear gene, right, in some cases really a single gene where the particular mistake is known, that would be able to be repaired would be great. And then you can worry simply about the very, very large problem of how do you deliver the repair mechanism to the cells that are affected in a specific way and do it on target and not go off target and do it at the right timing and reach all the places and repair and then get your mechanism out of there so it doesn't do damage, right? I just lumped together a very, very large series of problems, but at least we know what they are, right?
Yeah.
In other cases, we can't say that's the simple case. The cases are much, much more complex. So this puzzles us as scientists, and it's a very, very big challenge for pharma. And the question is, how do you go about it? And that brings in another aspect of what CRISPR can do, which is so dramatic, and that is it can help us create new models for studying the disease. And it can do it in human tissue, right, that's derived from iPSCs, from cells that are made to become stem cells, regular cells from your body, made to become stem cells, and that then they can be grown in culture, and they can even be grown into organoids. And they make a fantastic model that can be studied in very large scale and with great parallelism, and they can be engineered to contain different kinds of things that can act either as a potential repair mechanism in an assay for a mechanism of treatment, or they can be for fundamental research. For example, let's just take one of these examples. You have 90 genes that are responsible in some way that we don't really understand for an ultimate condition. And we understand something about the pathways that they are involved in. And that's the level of current analysis. You have a set of genes and they fit into a pathway, and you think that tells you something, but you don't know how these proteins that the genes encode interact in the cell in a specific compartment of the cell that gets affected. For example, a synapse between two neurons, which is so important for learning and memory and cognition and would be affected negatively in Alzheimer's. So imagine a case where you can engineer the system in order to be able to pull down groups of these proteins and ask how they interact. It's something that you could never do otherwise. You can't do in a human. You do it in a mouse. The mouse is not the right model. You can't look at the right, you usually look at one protein at a time or one gene at a time. And suddenly you can bring together these protein networks or these gene networks and look at how they really work together mechanistically. So CRISPR gives us the opportunity, right, to be able to get at design of models for understanding molecular mechanism in a way that we can never be done before. And that can then lead maybe to a CRISPR cure in the future. This is what really interested Roche and Genentech in part, right? That's part of what made for this great alliance for us. And it offers a great horizon.
Yeah, thank you for that. So maybe I can turn it over to Jennifer and ask her to talk a little bit about more about the CRISPR-Cas technology itself, as it seems like it will be a central part of this effort.
Thanks, Melinda. And it's a real pleasure to be here with all of you today, especially with my colleagues, Steve and Udi. As they very nicely described, I think we have an extraordinary opportunity right now to bring the CRISPR technology, which is a tool for gene manipulation, for genome engineering, to bear on questions about the actual mechanism behind neurodegenerative disease, as well as to actually impact the genetics of those diseases in ways that will have a real impact on patients. So my role in all of this, you're seeing behind me the Innovative Genomics Institute building at Berkeley. And this has been from the beginning a partnership, closely partnering with UCSF. And as a scientist who has worked for my career so far on developing fundamental understanding of biological systems, and that led us to the CRISPR technology, I'm incredibly excited about the opportunity to work with Steve and Udi and all of our colleagues to now bring this technology to bear on neurological disorders. And as you heard very nicely from Udi, one of the transformative aspects of the CRISPR technology is the fact that not only is it a great tool in the laboratory for understanding genetics of disease, but it also is a technology that can be used as an actual therapy or even a cure, if we can figure out which genes to manipulate, and importantly, how to do the manipulation, in what types of tissues, whether it's the brain or somewhere else in the body. We need to figure out how to do editing in ways that will be safe and effective, and importantly, ultimately widely available and accessible and affordable to people that need it. And that was really the founding idea and mission behind the IGI, was to develop genome editing in ways that will ultimately be available to as many people as possible. So I'm very excited to be part of this partnership for all of those reasons.
So maybe I can follow up on what you just said around, tissue specificity being an issue. So it seems that the delivery of the gene editing therapies to specific tissues can be a big hurdle. And I've heard that ferrying the CRISPR machinery into an organ as well protected as the brain is a major challenge. So maybe either Jennifer or Udi, you can talk a little bit about how that is being addressed.
Who's going first, Udi?
You, Jennifer.
Well, I'll say a little bit, and then I'm going to turn it over to the expert. But basically, we've been thinking about this question, the challenge of delivery, in the context of a number of different tissues, including the brain. Of course, it is a very challenging target. The brain is designed to keep things out for the most part. And so as scientists and technologists and clinicians, we have to figure out how we circumvent those natural mechanisms. But I think you couldn't really ask for a better team here to address this challenge, not only on the academic side, but also at Roche Genentech. We have experts who have been thinking about this problem for probably collectively decades. And we also have a lot of new innovators, students and postdocs that are working in our groups that are thinking creatively about how to address this challenge. And we've already made inroads that we're excited about in animal models. And I think over the next few years with this partnership, we have an opportunity to actually develop this in ways that will be clinically impactful.
So let me add, Jennifer's is as usual, unusually modest about, in this case, having, among other things, pioneered some novel approaches to doing gene delivery, which are actually very, very promising. The standard way, what's become a relatively standard way of trying to do gene delivery, is using viruses that have been re-engineered. And Berkeley is one of the places where there's been a lot of work on trying to do molecular evolution, it's called, to try to identify through very, very large screens versions of these virus coats that bring them to specific cells in the nervous system. That's something that actually is very, very promising. And yet there's a recognition in the general community, which includes the NIH Brain Initiative, that it's very, very important to develop new ways of doing the delivery that doesn't just rely on these viruses, because the viruses can, they're limited in what they can do. You have this little capsule that kind of looks like a space capsule, and what you rely on is like its outer surface. It should have some kind of specificity of what cells it sticks to. There's a certain limitation likely on how much you can change that around in order to give you specificity. So there are other ways that you want to do it, and there are ways that you want to be able to deliver cargo, meaning genes of various kinds of sizes that are hard to fit into these little space capsules. So there are other methods being done, and some of them are being pioneered now at IGI that are very, very promising. So I think that this is going to be one of the major frontiers, right? That you can solve every other problem, but if you can't deal with the delivery issue, then you're going to be stuck, right? So this is a major, major emphasis.
Okay. So maybe, Steve, I can bring you back in here. So, we've seen a high degree of failure in clinical trials to treat multiple neurological diseases. Why has that been such a big challenge? Is it for some of the reasons that have already been discussed, or are there additional reasons?
I think, as you've heard, these are difficult problems. Udi mentioned a really important one, which is our optimism that with better models, we will have more accurate representation of disease in a test tube or in an animal. And iPS cells and organoids in that area where we can truly make a human model of disease in the laboratory will be key, because as I said before, I think the over-reliance on inadequate models has been part of the difficulty in the neurosciences. I think that there are a few other things that we probably need to improve upon. We need to make our clinical trials much more efficient. And we need to make sure that our clinical trials are linked to the serious science that can really let us develop biomarkers, understand pathways, and that those need to be very transparent and always in the public domain, which it hasn't always been. We need better biomarkers. We have a responsibility to arm drug developers with endpoints that don't require thousands of patients studied for 2 to 3 years, and endpoints that accurately predict an outcome that's clinically meaningful. And I think this is why through NeuroHub, better neuroimaging and other biomarker initiatives are so high on what we really hope to do. And then also, I think we need regulatory agencies to help us and be more flexible in letting us choose biomarkers, especially for early studies, and maybe be flexible in terms of level of evidence that is needed before there's approval. So I think there's a lot that can be done on all sides. But one of the things that IGI and NeuroHub and ATN is doing writ large is bringing siloed communities together. And I think if we can bring together scientists like Jennifer and Udi with the most motivated clinicians and patient cohorts that we have, with medicinal chemists and drug development people, that creating that kind of community where we interact daily is another way to speed the inefficiency of current trials.
Mm-hmm. So maybe, Udi, I can follow up with a question there regarding translation. So that seems like also a difficult, path. I know that you started a company to use gene therapy to reverse a form of genetic blindness. How did fundamental research factor into that success story? And do you think that there's some lessons learned that are applicable to this context?
Yeah, I actually do. I think that first of all, it was a very unusual granting mechanism from NIH. This was part of the what was called the Roadmap Initiative that was started by Zerhouni when he was head of NIH. And what the concept of this component of it, which was called the Nanomedicine Development Centers, was there was an enormous advancement in material science that was called nanoscience at the time, right? It had to do with creating these tiny little weird molecules that have interesting physical or chemical properties, and they seemed to be ready to revolutionize maybe the way you make displays or wearable displays for our computers or something, or extremely thin material that could be used to give new properties to macro material. And the question was, could you ever turn any of these things into something that would actually be functional in medicine? And that was a really, really interesting and difficult challenge, because most of these things are actually the kind of things that you have to wear an astronaut suit in order to work with, because you don't inhale it, right? How would you ever get this to work in a biological setting, much less in something you could apply in the clinic? So they decided that they were going to actually say, okay, we're going to give it a 10-year run in order to produce something that is completely new. So you're going to have to work on something that is logical, potentially biocompatible, eventually applicable to some medical condition. And you have to have a plan for that right from the beginning, even though you're coming from way out of the normal field that's applied in medical research. So that was how we started. In our case, we started with a small chemical that is sensitive to light that we were using to attach to proteins to make them sensitive to light, right? And the idea initially was we could study the nervous system this way, as the kind of the way you do with optogenetics, except now you can make every protein that you want in the nervous system sensitive to light, and you can ask, what is it doing? Turn it on and turn it off, prevent it being turned on normally. That was what led to the beginning of this. It was really interesting for studying circuits and molecular interactions at synapses, and it wasn't clear how it would ever be involved in applied to a neurological disease, right? So we applied and we did not get in, right? So we came back a year later, and this time we came back with something where you said, okay, at least light reaches one part of the body, and that is the eye, right? So let's see if we can apply it in the eye. And we brought in a new team, which was interested in trying to do a retinal prosthetic, basically, after blinding diseases that are produced by degeneration of the photoreceptor cells that enable us to see light. That was my experience, but I think it's really relevant to us here too. We're starting in a place where we're trying to reach something that has been very hard, decades, as you said, right, of failure, not because of not trying, but because of the great difficulty of what's in front of us. We need to bring to it things and people and disciplines that are very, very different from the usual ones, right? It can't just have people who work on Alzheimer's with Alzheimer's patients. You need people who come from parts of biology that don't have anything to do with neuroscience, but who understand where things go wrong in a cell, right? That you could maybe work with, understand the mechanism of, or re-engineer potentially. You need people from engineering and the device engineering and from computer science in order to be able to bring new methodologies in. Actually, I'm very excited. I was just talking to Fyodor about this today, about the idea of bringing advanced kinds of machine learning and computation to how CRISPR is both designed and analyzed, so it can kind of reach a new generation of how CRISPR technology is used, right? These are things that are very, very non-standard, but bringing these things in will really, really strengthen the whole enterprise, I think, and they can produce the breakthrough. But what you need for that is a long run. You need the right kind of partnerships, and Roche Genentech really are taking the long view and ready to do that here, right? And you need to have what we have so well between the two campuses of UCSF and Berkeley and the IGI that bridges between them. You need to have this real depth of things that go from the clinic and all the way through the basic sciences, engineering.
Yeah, thank you for that. So, Jennifer, Udi mentioned a new generation of CRISPR technology. I know one of the newer CRISPR-based technologies that you think will be powerful for treating neurological diseases is something called epigenetic editing. Can you tell us a little bit about editing the epigenome and how that's different from traditional genome editing?
Well, first of all, you're right, Melinda, that CRISPR can be used in different modes. And as you just mentioned, in the mode where it doesn't actually cut DNA and lead to permanent changes to the genetic sequence, you can actually use it in a way where it is being employed to activate, increase, or decrease expression of certain parts of the genome. For example, up or down regulate output of particular proteins. And there's a lot of evidence in different neurological disorders that that's exactly the kind of manipulation that could be beneficial, both for understanding disease, but also importantly for treating it. And the thing we like about epigenetic manipulation of the genome is that it doesn't lead to a permanent change to the DNA sequence. It could potentially be safer. However, this mode of CRISPR has not yet been used clinically. So one of the goals of this partnership is to figure out, first of all, how could we use it in that mode for treating neurological disease? And then secondly, how do we do that in a practical sense? Again, it gets back to the question of delivery, as well as figuring out the right gene or sets of genes that would need manipulation using an epigenetic type of a tool.
Thank you. So let's maybe switch from talking about research, talking about delivery, more delivery to patients. The big question on everyone's mind is, when would a patient with a neurological disease expect to be able to access these new therapies? So maybe a round robin on some thoughts on that. Udi, you can go first.
No, I should go last. Steve should go first.
Oh, Steve, you're on mute.
Yeah, Steve's the doctor.
I have the most experience in this area, so my guesstimate will be the least worthwhile. It's better to ask Udi. These are difficult problems, and I think one of our challenges and the opportunity before us that we have to make the most of is to select the challenge, the problems, the clinical problems that we really have a chance to make an impact in, and that also will teach us as we go into patients the lessons that we need to learn for the larger diseases. So if Udi, you were right that I didn't mention Parkinson's, I didn't mention Alzheimer's, but the small proportion of patients who have single gene penetrant causes of Parkinson and Alzheimer might well be the first kinds of patients whom we might consider for these sorts of therapies, and maybe the disorders that also have phenotypes in the eye, if that was our goal. And in fact, of course, those are some of the directions that we are working in. And the other huge advantage of what we're all trying to do together is to recognize that by going back and forth between patients and the laboratory on a daily basis, that's how we're likely to make the progress most rapidly. It isn't bench to bedside only. We learn just as much from bedside back to bench. When we test our ideas, it's the bedside where our ideas are really road tested for their utility. So the kinds of communities that we're speaking about, also with very, very good people, which of course is the key, and making the small.
Steve, you put yourself on mute again accidentally.
I'm sorry.
It's okay.
Making the right decisions about which application makes the most sense in March 2021 is I think our challenge and responsibility, and what makes this also exciting.
So let me Steve was very good at not answering your question, you realize, right? And there's a reason for that, and you know what the reason is. And I think that the best answer can come from Jennifer, right? Because she can talk about something that's not neurological, but where CRISPR is already, being applied in clinical trials that she's involved in. And she can tell you both why that is the first place that she turned and where the challenges would lie for something else that could take much longer.
Yeah.
Right. Well, I think you're referring to sickle cell disease. So this has been one of the early targets of CRISPR in clinical trials. And for those that are following the news on this, you know that there already have been some very promising indications that CRISPR will be highly effective for this type of disorder. Sickle cell disease is in some ways, almost an optimal type of genetic disease to apply CRISPR to, because it is a disorder that involves a single gene. We know exactly what the genetic manipulation is necessary, what genetic manipulations are possible that could mitigate the disease or even cure it. And those are possible to institute with CRISPR. And then thirdly, it's a disease that affects the blood primarily. So it's possible to do the editing ex vivo by taking out blood stem cells from a patient, doing the editing in the laboratory, and then reintroducing the cells, the edited cells. So the whole challenge of delivery is kind of bypassed in that sort of a setting. So, we're excited at the Innovative Genomics Institute to be involved in this. We have a trial in sickle cell disease that's beginning this summer. And we're also hoping to take what we're learning from that effort and apply it to other diseases, including the ones we're discussing here. And again, it sounds like a tall order to go from the blood to the brain. On the other hand, I think that as you said, Steve, the more that we learn, especially in the clinic from patients, as we start to apply these kinds of tools, that does feed back into the laboratory to help educate scientists about, how the technology needs to advance. And that's absolutely what we're hoping to do in this partnership. So I'm also not answering your question, Melinda, about how long is it going to take. I think it's very hard to say, but we, rest assured that we're all, I think, very, very excited about the opportunities ahead of us. We want to be sure that we're being safe, first and foremost, and effective. So we don't want to rush things, but we also want to be moving together in a focused way towards the kinds of technologies and then their clinical use that will be truly impactful in patients.
Yeah. So
Linda, I might also just want to add, since we're a public, since this is a public conference, that it's important to remember that new technologies always take time for us to understand how to use them appropriately and most efficiently. So we've seen this with the genetic revolution, understanding genes and disease, with MRI technology. We've seen this with almost every new technological advance. So I think what the public also needs to is to understand is that patience and optimism is going to be needed, even if the tempo of immediate applications are not as fast as any of us hope they will be.
Mm-hmm. Yeah, I think that's a really, really good point, Steve. So, alongside the conversation around safety and efficacy and timing, there's also a consideration around equitable impact on society, equity, access. New technologies tend to be extremely expensive. And I think the last year has really highlighted to the public issues around equity and access as it relates to healthcare, right? So what are those considerations in conducting this research? And are there any ethical considerations around that as well? So I know this is another difficult question, so maybe Steve, you can kick us off.
Gee, I would think that Jennifer is in the best position here, but maybe I'll just say that safety, as Jennifer said, is foremost. Usefulness is foremost also. How do we think about very expensive opportunities that have marginal, if any, effect? This is a huge problem in the healthcare drug development industry.
Mm-hmm.
Dual use for good and bad purpose. Science will always be ultimately transparent and will have the potential for not only well-meaning ill use, but also ill use on its own. And we have to be very alert to that as well. I think that we also are going to have to deal with the problem of where does disease begin and tendencies end, and where does disease correction turn into enhancement of normal function. So there are, as you've said, there are all of these use questions. But I think that, as you've suggested, Melinda, the foremost one is, if my kids can get this, my neighbor's kids need to get this also.
Yeah. Yeah. So Jennifer, do you want to follow up on that? I think, this is a core tenet of the IGI.
Absolutely. I think that you said it very, very nicely, Steve. And, I think that's so important with a new technology like CRISPR, is to be thinking even now, even as we're in the early stages of developing it, how can we make sure that ultimately it becomes widely affordable and available? Because, many people have asked, why do this in a nonprofit setting at all? Like, why aren't we all just starting companies to do this work? And, I think there's a few good reasons for it. One is that it is still very early days with the technology for these indications. But secondly is, at least for me, is that I think there's a real need to be using a lens of equity when we think about how the science advances. And I get personally very motivated by that. And it's not something that necessarily works in a for-profit organization. However, it could certainly work in the context of the partnership we're talking about here with Roche Genentech, where yes, they are a commercial partner, but they are. We have some very clear alignments around how the technology will be developed fundamentally, and then how we're going to be able to deploy it later. So just very, very important I think to have this idea in front of all of us as we continue to advance the CRISPR technology, especially with an eye towards treating or eventually curing neurological diseases.
Yeah, thank you. So I think at this point, I will switch over to questions from the audience. So let's see. So maybe we can start with this one for, who would this go to? So the question says, I know we can use CRISPR to enhance the efficacy of analytical techniques, but can we also use it to test for possible unknown side effects of treatments? Maybe Udi would be best
that should be
placed.
Jennifer, but I'm happy to give my opinion. Yes, absolutely. The beauty of the system is that you can use it entirely to address things of that kind, right? Remember, you can use it in order to try to edit a target gene that is responsible for a disease, to repair it so that the disease-causing alteration is removed. But you can also use it as a way of engineering cells and tissues in order to have something that allows you to analyze how they work under conditions of another kind. You can do it in cells and tissues that come from a patient who has a particular condition, where you may or may not actually know what the cause of that condition is, right? And then you can use CRISPR to make alterations that allow you to, let's say, grab sets of proteins and look at how they're altered. If you did that, it's massively powerful analysis of a kind you normally cannot do to see what is altered in the patient or what is the effect of a drug treatment. So there are many combinations of that kind that are very, very powerful. And there are all sorts of screening projects that can be done, some of which are being done already in IGI, to try to design methods to give you more levers that you can use to control how genes get expressed, whether they get expressed more or less, specifically in certain tissues or others, that can give you even more power and then actually work eventually not only in a cultured system, but also in an organism.
So there's a follow-up question that I think relates to this from someone in the audience. They were asking whether epigenetic editing is targeted at a specific organ or is it done on the entire body?
Well, it could go either way. I think that for the kinds of things we're talking about here, ideally, we'd love to have it targeted to the CNS, to the brain, to the cell types that are specifically affected in a particular disorder, but that's a big challenge. And I think that the other thing to think about is that right now, as Udi alluded to, we have some technologies for delivering to the brain, but they involve either using viruses, which have various risks associated with them as well as cost, or using a more direct injection across the blood-brain barrier that, involves surgery and challenges there. So I think we just have to, as the field advances, we just have to continue to really think hard about exactly what is going to be the better way to deliver CRISPR, and it'll be very much dependent on the particular indication, whether it's important to be going after particular cells or cell types or even regions of the brain, let's say, or whether a more systemic delivery is going to be desirable.
Mm-hmm. Thank you.
Let me just add one thing, and that is that the amazing thing about this technology is that it's developing so quickly and its power is so high that you can ask the kind of question you just asked, Melinda: do you target or do you not target it? Or the questioner asked, right? How you target and what the cell types actually are of the brain is an active area of investigation right now. A lot of the work's been done on mouse, right, as a stand-in for us, and now there's a big turn to try to do it more in human, right? One of the strengths of our collective between UCSF, Berkeley, and University of Washington, which is all NeuroHub, are the brain banks that exist at UCSF and UW. And the analytics that's done on those brain banks are very, very powerful ways of trying to understand how many different cell types there are, what makes a cell type in the brain, and then eventually, which cell types are affected by a particular condition that then you would want to target. Mm-hmm. So we're actually the technology for the manipulation and analysis that CRISPR represents is being developed at the same time as all of this understanding of cell type and how the cells are connected and how they speak to one another, right, in other efforts. And you can imagine how this is just a massive amount of data to consider, which is why analytics and computer science, AI, and machine learning are going to be playing such an important part.
Wow. So we have a question from YouTube that's asking about exomes as a delivery vehicle, or identifying features of exomes that help them naturally cross the blood-brain barrier, and the potential to engineer synthetic vehicles from the bottom up.
Yeah, so I mean, I think that's a great idea. So these are secretions by a cell that contains stuff, and that stuff can be interesting stuff. It can be proteins and RNA, right? And of course, RNA is encoding, right? Jennifer's the beauty of Jennifer's work. We sometimes forget, where Jennifer comes from, right? But she comes from the world where RNA is king. We used to think that everything else was king, but RNA, remember, combines genetic information with the ability to do work, right? Acting like enzymes. And so the information transfer between cells that's represented by this is something that's interesting to try to consider as a model, either as a metaphor or actually as something to directly engineer and use. And in IGI and in Jennifer's own work, they're motivated by that to try to look at other ways of delivery, which could end up being very, very powerful. Why am I answering for Jennifer all the time? You know what? I'll tell you why, because I love the work, so I really want to talk about it.
I feel like you guys are going back and forth fairly well. So there's a more general question from Michaela. She's asking what neurological disorders are being looked at as the first to treat with CRISPR and why, and sort of what aspects make them good candidates for being the first. Jennifer or Steve?
That's you, Steve.
Well, I mentioned a few at the very beginning.
Yeah.
I think that the beauty of the sickle cell model that Jennifer mentioned is that there's a single building block of a single DNA molecule that is altered in almost everybody with sickle cell disease, and we understand that pathway well. And it's contained in a cell that is accessible and even can be removed and put back in. So there are many advantages to that kind of model. And understanding in the nervous system and in the panoply of diseases that we have, which models would most fit our ability to introduce the corrective. And as Udi said, either taking advantage of evolution to teach us how to efficiently incorporate extracellular molecules into cells is one superb way of leading in this research. But I think single gene disorders that reliably produce disease. Of course, we're interested in serious disorders and that are important health problems, and also where we understand enough about the course and the biology so that we can see if we're having an effect.
So there was a question really specific to Parkinson's, or a few questions actually. Could that be one of the disorders that you were talking about? People are asking what's the underlying cause and whether genome editing might be an approach.
So Udi had mentioned Parkinson's at the very beginning, and you may want to say something. It looks as if the person who wrote has one of the very rare and unusual changes in Parkinson's, which is not just the kind of building block we were speaking about before, but a gene that duplicated so that there are 3 copies instead of 2. And that's a slightly different genetic question, but maybe one that is also could also be considered as a target. Udi, you may want to speak about, there are 16 or so genes that we know about that cause Parkinson's disease. They're not all the same. The tissue damage is different, even if the patients look mostly the same. So this is another very important reason to connect with the clinical world with the genetics world.
Yeah. Let me maybe I'll answer in this way, which I hope it doesn't discourage people, but I want us to remember what one of the beauties and great complexities of treating neurological conditions is, and that is that in the nervous system, it's not like you just have 3 different cell types and one layer does this and another layer does another thing. Here you have cells that are hundreds of different cell types of neuronal cells interacting with several different cells that are non-neuronal called glia, some of which operate like the immune cells of the brain, and they are wandering around and doing the kind of things that immune cells do, but also interacting in ways that immune cells don't necessarily always do elsewhere in the body. And the health of every cell is very much dependent on all the cells that it interacts with. So if you have a condition that compromises the health of a particular cell type, then it's not clear. You have to expect that that's going to project from that cell to all the other cells that it's connected to in some way. So for some conditions, maybe that's not so confusing. Other conditions, it can be very, very complex. Mm-hmm. The reason I say this is because this is part of the challenge with Parkinson's. In Parkinson's, we know that cells that make dopamine, the neuromodulator dopamine, that are in the basal part of the brain, the basal ganglia, die off. And it's the loss of those cells that's responsible for there being less dopamine in the places where you need in the brain in order to maintain the normal functions. We usually think about motor coordination and initiation of movement, but in fact, dopamine plays a role in appetite and sleep and addiction and reward and many, many other things. And yet only some of the systems are affected strongly and some are affected much less. And the reason appears to be that not all dopamine-making neurons are equally susceptible and die off as much. We're still trying to understand that, right? They connect to different places. That may be part of the explanation, but they also have different cellular types, subtypes. So to really, this is just a very big part of the challenge of treating the conditions of the nervous system, so that even if we know that you have a single gene involved, let's say, for some of the patients with Parkinson's, it's a known gene, like Steve was just saying, and cells that are susceptible, it's not always clear exactly, how you can target just those cells at the right time, or whether you have to do other things in addition. And so these are the challenges. That doesn't mean that it's not doable. It's just telling you that we have to pay attention to a lot of extra things when you're dealing with the diseases of the nervous system.
Yeah. Thank you so much for that, Ehud. I think we only have 1 minute left, so we're almost out of time. This has been really fun and informative. I was wondering if maybe you can give me a 1 or 2 word of, what scientific innovation are you most excited about in the next coming year? So maybe Ehud, you can go first.
It's very easy for me. It's 2 words: Jennifer, Doudna.
Jennifer?
Delivery and brain.
Okay. And Steve?
I think medicine will be neuroscience, neurologic and psychiatric disease. That is the next place where the big things are going to happen.
Well, thank you all so much for your time, and thank you to the audience for joining us today. I hope you enjoyed the discussion. We will be posting a video of this discussion at the Innovative Genomics Institute website following the event. I'd like to thank our panelists and also our communications and public impact team staff at the IGI for organizing the event. I hope you all have a wonderful afternoon or evening, depending on where you are, and please visit our website for any upcoming events. Thank you all.
Bye, everybody.
Thanks, everyone.